Hi everyone, welcome to another episode of the Bio-Inorganic Chemistry course. Today we are continuing with the photosynthesis. In the last lecture, we have learnt about the various stages of photosynthesis, that is the light reaction and the dark reaction. So, we have to study in detail what are light reactions and the dark reactions. In that, the light reaction we have discussed in the last lecture, and in this lecture we have to continue a bit of light reaction. That is, light reaction is being further categorized into cyclic photophosphorylation and non-cyclic photophosphorylation. And then we will see what is dark reaction and what is the mechanism of dark reaction. To begin with, we start with the cyclic and non-cyclic photophosphorylation. Cyclic photophosphorylation always happens in photosystem 1. What is photosystem 1? I hope you remember, it is P700, that is where the cyclic photophosphorylation will occur. In cyclic photophosphorylation, the only product is ATP, while the non-cyclic photophosphorylation occurs
in both the photosystems 1 and 2.
What are the byproducts of non-cyclic photophosphorylation? We have NADPH produced, and also ATP is being produced in the non-cyclic photophosphorylation. Here you can see the representation of the non-cyclic photophosphorylation. I already mentioned that it contains both the photosystems, that is the photosystem 1 and the photosystem 2. We will see in detail how this photosystems are working in the coming slides. So, you should understand that in non-cyclic photophosphorylation, its name is coming due to that the flow of electron is non-cyclic in this photophosphorylation. So, we call it as non-cyclic photophosphorylation. The electron transport etc. we will see in the coming slides in detail. The end products of non-cyclic photophosphorylation are NADPH, ATP and also oxygen is evolved. What is the site of non-cyclic photophosphorylation? It occurs in the thylakoid membranes. Okay? It occurs in the thylakoid membranes of the higher plants. Okay? All the higher plants have the non-cyclic photophosphorylation mechanism. So, I already mentioned that non-cyclic photophosphorylation has the two photosystems functioning, that is both the photosystem 1 and photosystem 2, that is the photosystem 680 and the photosystem 700. So, what are the photosystems? Photosystems contain basically the chlorophyll molecule along with the accessory pigments. Here is the representation of the process. Here you can see both the systems, photosystem 1, photosystem 2. Photosystem 1 is having the maximum absorbance at 700 nanometer. Photosystem 2 is having the maximum absorbance at 680 nanometer. So, both the photosystems work together in non-cyclic photophosphorylation. Here in photosystem 2, we have already learned that during the light reaction, there is splitting of water molecule, and the electrons generated here are being transported along the electron transfer chain to the photosystem 1, and from there it is being utilized for the reduction of NADP to NADPH, that we will see in detail the mechanism in the coming slides.
Now, let us learn the steps involved in non-cyclic photophosphorylation.
So, first step in non-cyclic phosphorylation is the photolysis or the splitting of water by light. So, what is happening? We get electrons by the splitting of water, and this is kept within the photosystem 2, while the proton generated is left for the further use, and the two electrons that is generated is kept within the photosystem 2 itself. In the next step, what is happening? The photon absorbed by the chlorophyll pigments present in the reaction core of the system, that is, the photon is absorbed by the chlorophyll pigments present in the reaction center. That is the second step. The first step is the photolysis of water. In the next step, we have the absorption by the reaction center. We have the absorption of light by the reaction center. That is the second step of non-cyclic photophosphorylation. Now, we move on to the cyclic photophosphorylation. I already mentioned that cyclic photophosphorylation is exclusively performed by photosystem 1, that is the P700 photosystem. So, what is happening here? All the photosystems contain chlorophyll as the base and the other electron transport proteins here, and in cyclic photophosphorylation we have the byproduct only ATP. There is no NADPH production
in cyclic photophosphorylation. In non-cyclic photophosphorylation,
we have the production of ATP, we have the photolysis of water, we have the production of NADPH. But here in cyclic photophosphorylation, we have only the production of ATP. You can see the different electron transfer proteins and or charge carriers or electron carriers we have already studied in the previous lectures. We have the ferredoxins, we have the cytochrome complexes, we have the plastoquinone, etc. Now, let us see in detail what is cyclic photophosphorylation. Cyclic photophosphorylation primarily happens in the lower organisms. This is because it is being performed in the anaerobic conditions. Okay, it is being performed usually in the anaerobic condition. And we have high carbon dioxide compensation point. So, this is the condition for the occurrence of cyclic photophosphorylation. That is why it is occurring in lower organisms. And this is performed or in presence of photosystem 1. The cyclic photophosphorylation occurs in photosystem 1. So, what happens here? The electron flow begins in the pigment complex photosystem 2 will be passing from the electron acceptor plastoquinone, then to the cytochrome B6F in the mitochondria, then to the plastocyanin before returning to chlorophyll. So, that is the situation when both of these photosystems are coexisting. In that case, first the photosystem 2 will function, and from the photosystem 2 the electrons will be transferred to the photosystem 1, and there it will begin to function as photosystem 1 again. This is what is occurring in higher plants, but in lower organism there is only the functioning of photosystem 1. In the photophosphorylation cyclic 1, there is no production of NADPH or oxygen, and the NADP plus will not accept any electron and which is being sent back to the photosystem 1. The ATP is the only end product. Now, we have seen what is cyclic photophosphorylation, what is non-cyclic photophosphorylation, what are the photosystems, what is light reaction. Okay. Now, we are going to see how the photosystems capture light energy. In this diagram, you can see the light absorption by photosystems. You can see the reaction center here. This reaction center will probably be a chlorophyll molecule, and here you can see the other light harvesting pigments, that is antenna molecules. When light is falling, the antenna molecules will also involved in the light absorption, and it is being transferred, the photons are being transferred to the reaction center. So, what is happening there? The photosystems contain antenna complexes, and which is also having the chlorophyll as the reaction center, probably chlorophyll A or B, and other accessory pigments in the reaction center. Here in the photosystems, the pigments will be transferring the absorbed energy. The pigments are transferring the absorbed energy to the reaction center here. Within the reaction center, there is a special pair of chlorophyll molecules that is coupled by proteins. This is the reaction center. Inside that we have chlorophyll molecules being coupled with proteins. When light is striking on it, the electrons get excited into higher energy levels, like the light energy is being jumping on a trampoline and sending the electrons up in air, and it is handed over to the primary electron acceptors. This is how it is working. That is, when the light is striking, the electrons are getting excited to a higher energy level, like the light energy is jumping on a trampoline, and it will then send the electrons up in air, and it is handed over to the primary electron acceptors. Now, let us see the functioning of the photosystems independently in detail. This is a picture depicting the photosystem II. Here you can see that the electron acceptors plastoquinone. In photosystem II, we have the splitting of water here, and from photosystem II, the electrons are being transferred to the electron acceptors like plastoquinone, and it is being sent to the next photosystem, that is photosystem I. We will see it in detail shortly. So, how does this work? How does the photosystem work? In this case, first we will see how does photosystem II work. The first step is the light absorption, that is, it will absorb light energy around this 680 nanometer, that is the first step. In the second step, we have the electron capture. So, what is occurring here in electron capture? The excitation of the electron will occur from a lower energy state to the higher energy state by the consequence of light absorption. Then what will happen? This excited electrons, very important, will travel down the electron transport chain, that is the ETS. The excited electrons will travel through the electron transport chain. We have discussed in the previous lectures what are electron transport proteins, etc. So, here they will function and they are transferring the electrons to the next photosystem or the region of requirement for the synthesis of the energetic biomolecules. Okay. Along the way, more protons is being pumped into the thylakoid compartment. We know that as a consequence of water splitting, we will have the proton generated and it will be pumped to the thylakoid compartment. Okay. Splitting of water and releasing of oxygen. So, by the splitting of water, there is also release of oxygen accompanying that. Okay, you know that. So, meanwhile, the photosystem lost electrons is replenished by photolysis of water. That is, whatever the photosystem is giving through the electron transport chain to the next one, it is being replenished by the further photolysis of water. The oxygen resulting from the photolysis is given back to the atmosphere. So, this is how the photosystem II functions. Again, another pictorial representation of photosystem II, how the electrons are being regained. Here you can see the photosystem II. The electrons are generated, it is given to the primary electron acceptor and it will move down the electron transport chain, and which is being given to the photosystem I, and where it is being utilized for the reduction of NADP plus to the NADPH. This is how the photosystem II functions. Now, we move on to the next photosystem, that is the photosystem I. We call it also P700. So, here we have another set of enzymes and electron transport carriers. Some of them will be same also.
Photosystem I, also known as plastocyanin ferredoxin oxidoreductase, is a protein complex that uses light energy to transfer electrons. It is
present in thylakoid membranes of cyanobacteria and chloroplast. I already mentioned that photosystem 1 is being functioning in lower organisms like cyanobacteria. So, here it is driven by or performed by plastocyanin ferredoxin oxidoreductase that is present in the thylakoid membranes. Okay, this is the enzyme used associated with the chloroplast, that is the plastocyanin ferredoxin oxidoreductase. So, how does it work? The first step is the excitation of the electrons in photosystem 1. The photo-excited electrons enter the photosystem 1 via electron transport chain set in the thylakoid membrane. So, at first the electron will enter the thylakoid membrane from the electron transport chain. Then the next step is the chemiosmosis. What is the process of chemiosmosis? The energy fall that is being through the electron transport chain is being harnessed to transport H plus through the membrane into the thylakoid lumen to generate ATP. So, this is the chemiosmosis process. Here the energy fall across it, electron transport is being harnessed for what? To transport the hydrogen for generating ATP.
the next step is the conversion of NADP plus to NADPH.
The excited electrons in photosystem 1 will further oxidize NADP plus to NADPH will be needed, which will be required for the Calvin cycle. Hope you are clear with the processes involved in the photosystem 1. This slide represents the working together of photosystem 1 and photosystem 2. Thus, here you can see it is called the Z mechanism because it looks like Z letter. So, here what happens is that I already mentioned that both the photosystem 1 and photosystem 2 work together in a particular fashion which resembles the letter
Z.
So, how does it work? Let us see. When light is absorbed first by the photosystem 2, the electrons present in the reaction center will get excited, which are eventually trapped by the primary electron acceptors. This hall is thus the hall that is created in the reaction center by the departure of the photo-excited electrons is further replenished by the electrons extracted from water through a cluster of iron manganese in photosystem 2. In this process, oxygen, which is released when 4 electrons have been removed from 2 molecules of water. Oxygen is released when the water is being splitted. Further, what happens? The light energized electrons travel to photosystem 1 through the electron transfer proteins like cytochrome B6F, which is an enzyme found in the thylakoid membrane that catalyze the transfer of electrons from plastoquinone to plastocyanin via the electron transport chain. During this process, the chemiosmosis occur where the transfer of H plus ion takes place across the thylakoid membrane via plastoquinone, which creates a gradient within the chloroplast that is being used for the production of ATP molecules. When electron enters the photosystem 1, it will wait until the electron is excited by another photon. The electron thus excited is captured by another electron acceptor, which will flow down the chain of the electron carriers to NADP to reduce it to NADPH that will then enter the Calvin cycle. Here you can see that after the reduction from NADP plus to NADPH, it will then enter the Calvin cycle. So, there is a series of reactions that is occurring. So, both the photosystems work together in photosynthesis, and we say it as Z scheme mechanism. In oxygenic photosynthesis, the path of electrons from water through photosystem II and the cytochrome B6F complex to photosystem I and finally to NADPH. This is the pathway of electrons which is occurring in the higher plants, that is through the through the electron transport chain which is involving B6X complex. It is finally getting into NADPH. When these sequence of electron carriers are plotted against the reduction potentials, we get the path of the electrons which look like Z, and it is called the Z scheme mechanism. So,
in summary, we can say that the Z scheme mechanism is a pattern that explains the path of electron transfer during the light reactions of photosynthesis, or we can say it depicts how both the photosystems 1 and 2 work together during photosynthesis.
so in summary, we can say that photosynthesis actually encompasses two processes. The first one is the light dependent reaction and the other is the dark reaction. So, in light reaction, what does occur? It will use the sunlight to synthesize ATP and NADPH. And in the dark reaction, what is actually occurring? It is the assimilation of carbon dioxide. So, here in dark reaction, the ATP and NADPH are utilized to reduce carbon dioxide to form the triose phosphate via a set of reactions which is known as the Calvin cycle. The ATP and NADPH is utilized for the reduction of carbon dioxide to form triose phosphates first, and then consequently it will form carbohydrates. Now, we are moving on to the next set of reaction or the next stage of photosynthesis, that is the dark reaction. Dark reaction is also known as Calvin cycle, or we can say it is the light independent reaction, that is light is not required in this phase. Where does it occur? This dark reaction occur at the stroma. What is stroma? It is the aqueous lumen layer in the chloroplast. Now, what are the requirements for dark reaction? We need the NADPH and ATP that is being produced in the light reaction. Another requirement is the Rubisco enzyme. It's very, very important. Another requirement is the Rubisco enzyme. It is the ribulose biphosphate carboxylase enzyme. We also require the carbon dioxide molecule. Here you can see the representation of the dark reaction. We have the carbon dioxide that is being fixed by the enzyme Rubisco. Okay, what is Rubisco? It is the ribulose biphosphate carboxylase enzyme.
Also note that Rubisco is a 5-carbon containing enzyme or compound. Here what happens is that the carbon dioxide is being fixed by the help of the 5-carbon Rubisco enzyme to generate glycerate phosphate first and then converted to triose phosphate. Further, the triose phosphate is being transformed into carbohydrates or glucose. Finally, the Rubisco enzyme is regenerated. Dark reaction can be divided into 3 main stages. The first stage involved is the carboxylation, followed by reduction, and the final stage is the regeneration. Now, let's discuss these steps in quite detail.
The first stage is the carboxylation stage. So, in the carboxylation stage, what is occurring? The ribulose biphosphate, which is a 5-carbon compound, is combining with carbon dioxide to generate a 3-phosphate glycerate, which is a 3-carbon compound.
Or simply we can say that carbon dioxide is being captured by the Rubisco enzyme. This is the first step in dark reaction.
The next step is the reduction. So, here what happens? There is a series of reaction that leads to the formation of glucose from the 3-phosphoglycerate stage to the formation of glucose. Here 2 molecules of ATP is used for phosphorylation, 2 molecules of NADPH is also used for
reduction of fixed carbon dioxide.
we are in this stage, that is the reducing stage, we are utilizing the NADPH and also ATP which was obtained from the light reaction. And the last step of the dark reaction is the regeneration. The regeneration of the RUBP enzyme. Okay. So, this require again one more ATP for the phosphorylation to regenerate RUBP. Hope you are clear with the steps involved in Calvin cycle. The photosynthesis process at a glance. So, what is occurring? There is the occurrence of split of water during the light reactions. Water is being split into we get proton and electron which is being utilized to
synthesize ATP and NADPH, which is further required to fix
carbon dioxide in the Calvin cycle.
And in Calvin cycle, carbon dioxide is transformed into glucose or carbohydrate. During the process of photosynthesis, plants take in carbon dioxide and water. The water is oxidized, losing electrons, while the carbon dioxide is reduced, gaining electrons. The process transforms the water into oxygen and the carbon dioxide into glucose. Or simply we can say that photosynthesis is the process by which plants convert light energy into carbohydrates or glucose, releasing oxygen as the byproduct.
So, hope you are all clear with the process of photosynthesis, the 2 stages of photosynthesis, that is light reaction and dark reaction. What are the processes involved in light reaction?
That is the cyclic and non-cyclic photophosphorylation.
And how is the Calvin cycle, that is the dark reaction occurring, the 3 stages that is happening in dark reaction, and the concept and the chemistry of photosynthesis have been discussed in this lecture. Hope you have all benefited from this video. Thank you.